One of the major approaches to understanding disease in the modern era is genetic research. Substantial funding has gone into building technology and knowledge focusing on genetics. Near the end of the last century, these efforts were celebrated when it was announced that we had successfully sequenced, read, the complete human genetic code. Now we are able to sequence, read, the important parts of any patient's genome, so-called whole exome sequencing, within a week or two, or even days if it is urgent. Yet this massive amount of information has not provided the tremendous insight that was promised. Why is this? The promise of genetic sequencing relied on several assumptions that have ended up not to be as solid as previously believed. ASD is a prime example of this. First, many diseases found to have a strong genetic basis are found to have an error in the code for one of the 24,000 genes on our 23 pairs of chromosomes. Now it is being realized that many diseases are not that simple. Like eye color, which is determined by multiple genes, it may be that some medical diseases may involve multiple genes that have no particular obvious relationship to each other or the disease itself. Given the large number of genes in our body, this becomes a large and complicated puzzle. Second, not all changes in the genetic code cause the development of disease by themselves. They may simply be risk factors that require another causal factor. Without identifying such factors, it is difficult to identify the changes in the genetic code that lead to disease. Also, many of these genetic changes are what we call polymorphism, indicating that they are relatively common changes in the genetic code that are possessed by many people without disease. Third, even with genetic disorders that are very closely linked to ASD, the association with ASD is not one-to-one. For example, Fragile X is the most common single-gene disorder associated with ASD, supposedly accounting for up to 5% of ASD cases. However, only 30% of cases of children with Fragile X have ASD. This theme is true for many other genetic disorders closely associated with ASD. Thus, there must be some additional factor that causes ASD in these genetic disorders. Fourth, there is good evidence that many diseases, like ASD, involve disruption of mitochondrial function. The mitochondrion is the only other part of the cell besides the cell nucleus, where the chromosomes reside, that carries genetic information. Mitochondria are exclusively inherited from the mother, so the inheritance of genes in the mitochondria is different from other genes in the cell, but the effects of mitochondrial dysfunction can be profound. Fifth, we are learning that genes are regulated through complicated molecular mechanisms. Some of these epigenetic processes rely on metabolic processes called methylation, which is known to be disrupted in ASD. Epigenetic processes are responsible for turning genes on or off, regardless of whether there are errors in their genetic code. Thus, standard methods for sequencing the genetic code to search for dysfunctional genes will not easily pick up genes that are silenced because of epigenetic processes. Sixth, like many developmental disorders, the errors in the genetic code identified in many children with ASD appear as what is called de novo, meaning that they are new mutations not found in the genetic code of the parents. Thus, these mutations arose during formation of the egg or sperm, or shortly after the egg and sperm combined to form the zygote. These de novo mutations are usually caused by errors induced into the genetic code, mostly by negative environmental influences. So even when genetic mutations are found to cause ASD, the ultimate culprit may not be genetic. In fact, it might be environmental. Finally, one of the reasons that ASD research focuses on genetics is that it has high heritability. For example, siblings are more likely to have ASD.